Welcome to Inert Chemistry. As you know, we started drug metabolism and chemotherapy. So, in this third lecture, we discuss about the mechanism of action of beta-lactam drugs. It is the third lecture of this series. So, let's begin. Here we see the mechanism of beta-lactam drugs. All the beta-lactam antibiotics like penicillin, cephalosporin, carbapenem, and monobactams, they work by inhibiting the cell wall synthesis or function. Means that these beta-lactam drugs, they stop the synthesis of cell wall of bacteria and thus control the bacteria. Here we see that how the antibacterial drugs inhibit the bacterial cell wall synthesis and its function. Different type of bacteria are broadly classified into gram-positive and gram-negative bacteria. Both these gram-positive and gram-negative bacteria have difference in the structure of their cell wall. Here in the first section of this diagram, a gram-positive bacterial cell wall is shown. It contains a cell membrane and an outer peptidoglycan. Peptidoglycan is a polymer that is made up of polysaccharides and amino acids. As the name shown, peptidoglycan, glycans mean sugars and peptido means amino acid. So it is a polymer that is made up of polysaccharides and amino acid. While in case of gram-negative bacteria, in addition to the cell membrane and peptidoglycan, there is an additional outer layer present. This is called as outer layer and it is present in gram-negative bacteria and missing in gram-negative bacteria. Here we have opened the structure of this peptidoglycan. This peptidoglycan structure is opened here, and as I have already described, it is made up of peptidoglycans and amino acids. NAM and NAG are the peptidoglycans. These NAM stands for N-acetyl glucosamine and NAG stands for N-acetyl muramic acid. Both these are polysaccharides and these orange colored dots are actually the amino acid that cross-linked these polypeptide chains. Transpeptidases are the enzymes that help in the cross-linking of these polysaccharide chains and thus strengthen the bacterial cell wall. So here we see that how these antibacterial drugs weaken this bacterial cell wall. The antibacterial drugs that mainly include beta-lactams. When we ingest the beta-lactams, then they enter into the bacterial cell through cell membrane. The cell membrane of gram-negative bacteria have additional layer and this layer contains porins present in it. As here also you see, these are the barrel-shaped organ or proteins. These are called porins, and the antibacterial drugs like beta-lactams They enter through these porins and cross the peptidoglycan chain and reaches to the outer membrane or outer surface of cell membrane. Here you see that these antibacterial drugs, they passes through porins, and through porins they reaches to finally reaches to these outer surface of the cell membrane. On the outer surface of the cell membrane, they attach to a specific protein. This protein is called penicillin binding protein. These beta-lactams, they attach to these penicillin binding proteins. Once they get binded, once they bind, they get activated. When these beta-lactams get activated, they again swim into the peptidoglycan, and here they again go back into the peptidoglycan chain, and here they inactivate the transpeptidase enzymes. And as you know, transpeptidase enzymes are involved in cross-linking. So these activated beta-lactams then inactivate the transpeptidase enzymes and they break the cross-linking. Here you see that this cross-linking is broken down. So when this cross-linking is broken down, the structure of bacterial cell wall is no more and it is broken down. Without the proper cross-linking, the bacterial cell wall becomes very weak and fragile. Due to the weakened cell wall, the internal osmotic pressure of the bacteria becomes unmanageable, and eventually the bacterial cell swell and burst. That is called lysis, and due to the internal pressure, the bacterial cell died. This is all about the mechanism of the beta-lactam drugs. Here you please read this slide carefully. It will be very helpful for you to write this question as a short and long answer in exam. See this diagram. It is the simplest diagram of the gram-negative bacterial cell wall. Here it contains this cytoplasmic membrane and a peptidoglycan chain and an outer membrane. This outer membrane contains porins, and this is PBP, penicillin binding protein.